For this assignment, we are asked to identify each step that is involved in generating acetyl CoA from pyruvate. Glycolysis is the breakdown of glucose by enzymes in which energy and pyruvic acid are released. Pyruvate oxidation must occur in order to prepare for the Krebs cycle. At the end of glycolysis, we are left with 2 pyruvate molecules. Each of these molecules contain large amounts of usable energy. Pyruvate oxidation acts as a key component to link glycolysis to the rest of cellular respiration by capturing this remaining energy in the form of ATP. Pyruvate oxidation is considered a preparatory step necessary to enter into the Krebs cycle. The purpose is to convert pyruvate, a 3-carbon compound, into acetyl CoA, a 2-carbon compound. Acetyl CoA then acts as fuel for the next stage of cellular respiration. Pyruvate itself is produced in the cytoplasm via glycolysis. The process of pyruvate oxidation occurs in the mitochondrial matrix, assuming we are discussing its function in eukaryotes. This chart here shows the steps of pyruvate oxidation. In step 1, each pyruvate loses a carbon atom with the release of carbon dioxide. In step 2, we see that electrons are transferred to NAD+, meaning NAD+ is reduced to NADH, which will then be used by the cell to produce ATP. In step 3, an acetyl group is transferred to coenzyme A to produce acetyl CoA. Acetyl CoA then binds with oxaloacetic acid, a 4-carbon compound, where it transfers its acetyl group to oxaloacetate to go on to form citrate, necessary for the citric acid cycle.